As the outer world gradually quiets and the larger motions of the day begin to slow, it can be comforting to turn attention inward, toward a realm that is always active, yet never hurried. Beneath the surface of thought, beneath breath and heartbeat, there exists a vast molecular landscape where countless processes unfold with remarkable calm. This is not a place of urgency or noise, but one of steady rhythm and gentle order. It is here, in the softly lit interior of the cell, that proteins quietly take shape. Proteins are among the most familiar yet least noticed participants in life. They do not announce themselves, and they rarely draw attention, yet without them nothing living could persist. Every movement of muscle, every chemical reaction, every moment of repair or renewal relies on these molecules performing their roles with quiet consistency. Even now, as the body rests, proteins are being formed, adjusted, folded, and guided into place, continuing their work without interruption. To understand protein folding is not to confront something mechanical or rigid, but to observe a natural settling process, much like the way water finds its level or how mist drifts into valleys at night. Folding is not imposed upon proteins from the outside. Instead, it emerges from the nature of the molecules themselves, shaped by simple physical principles that operate gently and predictably. There is no urgency in this process. There is no sense of striving. There is only movement toward balance. At the molecular scale, life unfolds in silence. Proteins are assembled within cells, surrounded by water, ions, and other molecules, all sharing the same small space. This environment is warm, fluid, and crowded, yet remarkably organized. Within it, long chains of amino acids begin their quiet transformation, bending and curving until they reach shapes that allow them to function. This transformation, known as folding, is one of the most fundamental acts in biology, repeated endlessly across all living organisms. It may help to imagine this molecular world not as a factory, but as a slow-flowing stream. Molecules drift, encounter one another, separate, and meet again. Nothing is forced. Interactions occur because they are favored, because they reduce tension, because they lead to stability. Protein folding follows this same logic. A newly formed protein chain does not search anxiously for its final form. Instead, it gradually relaxes into it. The story of protein folding is deeply connected to the idea that shape carries meaning. Unlike many objects we encounter in daily life, proteins are not defined primarily by their size or composition, but by their three-dimensional form. The same sequence of building blocks, arranged differently in space, can produce entirely different outcomes. In this way, folding is not an optional step, but the very act that gives a protein its identity. Yet for now, it is enough simply to acknowledge that this process exists, unfolding continuously within every cell. There is no need to grasp the details all at once. Molecular biology does not demand alertness or effort. It invites patience. Like watching clouds slowly change shape, understanding can emerge gradually, without strain. Proteins begin their lives as simple chains, assembled one unit at a time. At the moment of their creation, they are extended and flexible, lacking the structure they will eventually adopt. This initial state is not a flaw or an error. It is a necessary beginning, allowing the chain freedom to explore the possibilities available to it. Over time, interactions between different parts of the chain and between the chain and its surroundings guide it toward a more compact and stable arrangement. This journey from simplicity to structure is guided by tendencies rather than commands. Certain parts of the protein are drawn toward water, while others avoid it. Some segments naturally align, forming repeating patterns, while others curve inward, sheltered from their environment. These tendencies arise from the chemical properties of the amino acids themselves, properties that have remained consistent across billions of years of evolution. What is striking about protein folding is how little oversight it requires. There is no central controller dictating the final shape. Instead, folding is an example of self-organization, where order emerges from interaction rather than instruction. Given the right conditions, the protein simply becomes what it is meant to be. As night deepens, it can be reassuring to know that this same principle applies broadly throughout biology. Life maintains itself not through constant vigilance, but through systems that naturally drift toward balance. Cells do not need to monitor every molecule actively. They rely on the quiet reliability of physical laws that guide molecules into useful arrangements. Even when assistance is needed, it is provided gently. Some proteins receive help from other proteins, known as chaperones, which ensure that folding proceeds smoothly. These helpers do not force or reshape. They merely provide space, protection, and time. The emphasis remains on calm progression rather than correction. In this first part of our exploration, there is no need to hurry forward. Protein folding is not a story with a dramatic beginning or a sudden revelation. It is a continuous background process, always present, always unfolding. By beginning here, with the simple acknowledgement of this molecular calm, we prepare the ground for deeper understanding later on. As the body rests, countless proteins are folding correctly, finding their shapes, and contributing to the steady maintenance of life. They do this without awareness, without intention, and without pause. Their quiet activity is part of what allows rest itself to occur. Muscles relax because proteins enable it. Thoughts slow because proteins support the cells that generate them. There is a certain comfort in recognizing that even at rest, the body is supported by processes that require no attention. Protein folding does not wait for consciousness. It continues through sleep, through stillness, through every quiet moment. In this sense, it mirrors the rhythm of rest itself, active, yet effortless. As we move forward in later parts, we will gently explore how proteins are built, how they fold, and why their shapes matter. For now, it is enough to simply remain here, aware that within every cell, an elegant molecular choreography is taking place. A choreography defined not by speed or force, but by balance, preference, and calm order. And so, as attention softens and the mind settles, this molecular night continues undisturbed, quietly sustaining life from within. 2. Proteins, as everyday molecules. Proteins are often spoken of as complex or advanced, yet they are among the most ordinary substances within the living world. They are present everywhere life exists, woven into the quiet fabric of cells, tissues, and organs. Far from being rare or exotic, proteins are familiar companions, working steadily and repeatedly in ways that support even the simplest forms of life. From single-celled organisms drifting in water to the intricate systems of the human body, proteins are always there, performing their tasks without ceremony. At their core, proteins are molecules built for work. They serve as builders, messengers, transporters, and regulators, carrying out functions that keep cells alive and balanced. Some proteins form the structural framework of cells, giving them shape and resilience. Others act as enzymes, gently guiding chemical reactions so they occur smoothly and efficiently. Still others carry signals, allowing one part of a cell or organism to communicate with another. Each of these roles depends not on complexity for its own sake, but on reliability and repetition. What makes proteins especially interesting is how ordinary their beginnings are. They are not born fully formed or specialized. Instead, each protein begins as a simple chain, assembled step by step from smaller components. This process occurs continuously, guided by instructions stored within genetic material. Yet the instructions themselves do not dictate the final shape and detail. They specify the sequence, and from that sequence, the protein's behavior emerges naturally. In daily life, we encounter proteins most often through food. When we eat, proteins are broken down into their basic components, and later reused to build new proteins within our own cells. In this way, proteins move quietly from one living system to another, recycled and repurposed over time. The protein that supports a leaf, a grain, or a seed may eventually become part of a muscle, an enzyme, or a signaling molecule in another organism. This continuity reinforces the idea that proteins are not confined to any single form of life, but belong to life as a whole. Within the cell, proteins are everywhere. They line membranes, fill the interior spaces, and attach to one another in temporary or lasting arrangements. Some proteins remain fixed in place, while others drift freely, interacting briefly before moving on. Despite this constant motion, there is no sense of chaos. The cell maintains a steady internal environment in which proteins can perform their functions with remarkable consistency. It is easy to imagine that such an environment would be tightly controlled or rigidly organized, yet much of cellular life relies on flexibility. Proteins are not static objects locked into place. They are dynamic, capable of subtle movements and adjustments. These small changes allow them to interact with other molecules, respond to changes in their surroundings, and return to a resting state when their task is complete. This flexibility is closely tied to protein folding. A protein's folded shape is not a frozen structure, but a stable arrangement that still allows for gentle motion. Like a resting body that shifts slightly with each breath, a protein maintains its form while remaining responsive. This balance between stability and movement is essential. Too rigid, and the protein cannot function. Too loose, and it cannot maintain its role. When we consider proteins as everyday molecules, it becomes clear that their reliability is more important than their complexity. Cells depend on proteins to behave predictably, to fold correctly, and to interact in familiar ways. This predictability allows life to persist without constant oversight. Once a protein is properly folded, it can perform its function repeatedly, often thousands or millions of times, before being replaced. The sheer number of proteins within a cell is difficult to imagine, yet their presence does not overwhelm the system. Instead, each protein occupies a role that fits naturally into the whole. Some are abundant, appearing in large quantities to support common tasks. Others are rare, present only when needed. Together, they form a balanced molecular community. What is especially calming about this picture is how little effort it requires. Proteins do not struggle to find their purpose. Their structure and function arise naturally from their composition and environment. When conditions are right, folding occurs. When folding is correct, function follows. This sequence unfolds quietly, without urgency or stress. Even errors, when they occur, are handled without alarm. Cells have ways of recognizing proteins that are no longer useful or correctly folded, and they gently remove them. New proteins take their place, continuing the cycle. This ongoing renewal contributes to the steady rhythm of life, allowing cells to adapt and persist over time. In this sense, proteins reflect a broader principle found throughout biology. Life does not rely on perfection, but on balance and resilience. Systems are designed to tolerate variation, to correct gently, and to move forward without disruption. Protein folding fits seamlessly into this philosophy. It is not about achieving an ideal form once and forever, but about maintaining functional shapes within a living, changing environment. As the body rests, proteins continue their everyday work. Enzymes guide chemical reactions that repair and replenish. Structural proteins maintain the integrity of cells and tissues. Transport proteins move substances where they are needed. None of this requires conscious awareness. It happens automatically, supported by the same physical principles that govern all matter. There is a quiet reassurance in knowing that life depends on processes that do not demand attention. Protein folding and function are steady, reliable, and deeply ingrained in the nature of living systems. They operate in the background, allowing larger experiences, movement, thought, rest to unfold smoothly. By viewing proteins as everyday molecules, we remove any sense of distance or abstraction. These are not remote scientific curiosities, but intimate participants in every moment of life. They are present in every breath, every heartbeat, every pause. Their folding and unfolding mirror the rhythms of rest and activity that define living systems. As this gentle exploration continues, we will gradually look more closely at how proteins are built and how their shapes arise. For now, it is enough to rest with the understanding that proteins are not rare or exceptional. They are constant companions, quietly sustaining life through their steady presence and dependable form. 3 amino acids, simple building units. At the foundation of every protein lies a small and unassuming group of molecules known as amino acids. They are modest in size and limited in variety, yet from them arises an extraordinary range of forms and functions. In many ways, amino acids represent one of biology's most calming ideas, that immense complexity can emerge from a simple, repeating set of parts. There is no need for endless variation at the beginning. A small toolkit, used patiently and consistently, is enough. Amino acids are present in all living things, whether in a microscopic bacterium or a towering tree, whether in muscle tissue or in the cells of the brain. These same molecules appear again and again. Life does not invent new building blocks for each organism. Instead, it relies on a shared molecular language, one that has remained remarkably stable over immense spans of time. This continuity gives biology a sense of quiet coherence. Each amino acid follows a common pattern. At its center is a small carbon atom, bonded to a few familiar companions. One side carries an amino group, containing nitrogen and hydrogen. Another side holds a carboxyl group, made of carbon and oxygen. These two features give amino acids their name and their defining chemical behavior. Attached as well is a single hydrogen atom and a variable side chain, often referred to simply as an R group. It is this side chain that gives each amino acid its distinct personality. Despite the shared structure, the side chains vary in subtle but important ways. Some are small and neutral, others larger and more complex. Some carry electrical charges, while others avoid water and prefer the company of similar groups. These differences are gentle rather than dramatic, yet they are enough to influence how amino acids behave when linked together. Over time, these small preferences guide the folding of proteins, shaping them without force or instruction. There are only 20 standard amino acids used by living organisms to build proteins. This limited number might seem surprising at first, but it is precisely this constraint that allows for reliability. With just 20 options, cells can produce proteins predictably, ensuring that the same sequence will fold in the same way each time it is made. This consistency is essential for life, allowing biological systems to function without constant correction. Amino acids do not exist in isolation for long within cells. They are brought together through a steady process that links them into chains. Each connection forms a bond between the carboxyl group of one amino acid and the amino group of the next, releasing a small molecule of water in the process. The result is a growing chain, flexible and extended, with its side chains projecting outward like small branches. At this early stage, the chain has no fixed shape. It is free to move, bend, and interact with its surroundings. This freedom is not disorder, but opportunity. The chain explores different configurations, guided by the chemical tendencies of its amino acids. Some parts are drawn toward water, others shy away. Some side chains attract one another, while others prefer distance. These interactions are quiet and continuous, shaping the chain gradually. It is important to understand that amino acids do not need to know the final form of the protein. There is no blueprint that describes the folded shape directly. Instead, the sequence itself contains all the necessary information. Given time and the right environment, the chain will settle into a structure that reflects the combined preferences of its components. This process is slow enough to be stable, yet efficient enough to support life. In daily experience, we might compare amino acids to letters in an alphabet. Each letter has its own form, yet meaning arises only when letters are arranged into words. In the same way, individual amino acids are simple, but when combined in specific sequences, they produce proteins with unique roles. The analogy holds further in that the same letters can be used to form countless words, just as the same amino acids can give rise to an immense variety of proteins. Within the cell, amino acids are continuously recycled. Proteins that are no longer needed are broken down. Their amino acids return to a common pool. From there, they may be used again to build new proteins. This cycle of construction and deconstruction contributes to the steady balance of cellular life. Nothing is wasted, and nothing is rushed. The materials of life circulate calmly, supporting renewal. The presence of amino acids also links nutrition to molecular biology. When we consume protein in food, we are not absorbing ready-made proteins for direct use. Instead, digestion breaks them down into individual amino acids. These amino acids enter cells and become raw material for new proteins, shaped according to the body's current needs. In this way, external nourishment becomes internal structure, guided by the same quiet rules. What is especially soothing about this process is its simplicity. Amino acids do not compete or struggle. They respond to their chemical environment in predictable ways. When conditions change, their behavior adjusts smoothly. This responsiveness allows proteins to fold reliably, even within the crowded and dynamic space of the cell. The side chains of amino acids play a central role in this reliability. Some side chains carry a slight positive or negative charge, allowing them to attract or repel one another gently. Others are uncharged but polar, interacting comfortably with water. Still others are nonpolar, preferring to cluster away from water's influence. These tendencies are not forces in the dramatic sense, but preferences that guide the folding process like a quiet inclination. As the protein chain begins to fold, these side chains find themselves in new relationships. Some form temporary contacts, testing stability. Others settle into positions that remain fixed for longer periods. Over time, a pattern emerges, reflecting the collective behavior of the amino acids involved. This pattern becomes the protein's structure, and with it, its ability to function. There is no urgency in this transformation. Folding occurs as part of the natural flow of cellular life. Proteins are given time to explore and adjust, supported by their environment and, when needed, by other proteins that offer gentle assistance. The emphasis remains on balance rather than speed. By focusing on amino acids as simple building units, we begin to see protein folding not as a mysterious or delicate process, but as an expression of basic physical principles applied repeatedly. Small differences accumulate into meaningful outcomes. Simple components combine to create functional forms. This is a recurring theme throughout biology, offering a sense of quiet reassurance. As rest deepens and attention softens, it can be comforting to remember that life depends on processes that are both simple and dependable. Amino acids have been linking together in this way for billions of years, forming proteins that support every living system. Their steady behavior underlies the calm persistence of life itself, unfolding gently at a scale too small to see, yet close enough to sustain us always. 4. Linking amino acids into chains. Once amino acids are available within the cell, their next quiet task is to join together, forming the long chains that will eventually become proteins. This linking process is steady and methodical, unfolding without haste. One amino acid at a time is added to a growing chain, each connection forming a bond that is both strong and flexible. Over time, these repeated linkages create a structure long enough to begin expressing the subtle tendencies that will later guide folding. The connection between amino acids is known as a peptide bond. It forms when the carboxyl group of one amino acid meets the amino group of the next. As they join, a small molecule of water is released, and the two units become part of a single, continuous chain. This reaction occurs again and again, producing what is called a polypeptide. The repetition of this simple chemical event is one of the most common processes in all of biology, yet it proceeds quietly, without fanfare. Within the cell, this assembly takes place at specialized molecular structures that read genetic instructions and translate them into amino acid sequences. The instructions determine the order in which amino acids are linked, but not how the final protein will look in 3 dimensions. At this stage, the focus is on sequence rather than shape. The chain emerges gradually, extending outward, flexible, and unformed. As the chain lengthens, it remains remarkably uniform along its backbone. The repeating pattern of atoms that forms the main chain is the same for every protein, regardless of its eventual function. This uniformity provides a stable framework, while the side chains of the amino acids project outward, introducing variety. It is these side chains, differing from one amino acid to the next, that will later guide the folding process. The newly formed chain does not wait until completion to begin interacting with its environment. Even as it is being assembled, parts of it may start to bend or align, responding to the surrounding water and nearby molecules. This early movement is gentle and reversible, allowing the chain to explore different positions without committing to a final form too soon. There is no pressure to finish quickly. Folding begins naturally when conditions are right. The direction of the chain also matters. One end of the polypeptide has a free amino group, while the other has a free carboxyl group. This orientation gives the chain a sense of direction, which remains consistent as it folds. Though this detail is subtle, it contributes to the overall order of the protein, ensuring that folding follows a predictable path. As more amino acids are added, the chain becomes long enough for different regions to encounter one another. Side chains that are distant in sequence may come close in space, testing possible interactions. Some of these encounters will be fleeting, while others will prove more stable. The chain adjusts continuously, responding to these internal and external influences. It is important to note that the chain is never isolated. It exists within the crowded interior of the cell, surrounded by water molecules, ions, and other proteins. This environment plays a crucial role in shaping how the chain behaves. Water, in particular, encourages certain side chains to remain exposed, while others retreat inward. These tendencies arise naturally from the chemical properties of the amino acids, requiring no guidance beyond the environment itself. Despite the apparent complexity of this setting, the process remains calm and orderly. The cell provides a stable temperature and chemical balance, allowing the chain to form and adjust without disruption. There is no moment of chaos, only continuous motion guided by simple rules. As the chain grows longer, it gains more opportunities for internal organization. Certain segments may begin to adopt repeating patterns, while others remain flexible. These early patterns are not yet the final folded structure, but they represent the first steps toward it. The chain does not rush to completion. It pauses, shifts, and settles incrementally. This gradual approach offers an important advantage. By allowing the chain to explore many possible arrangements, the likelihood of finding a stable and functional shape increases. Folding is not a single leap, but a series of small adjustments, each one guided by the combined tendencies of the amino acids involved. Throughout this process, the cell remains attentive, but not intrusive. When needed, helper proteins may briefly interact with the growing chain, preventing unwanted tangles or providing space for proper movement. These interactions are temporary and gentle, ensuring that the chain retains its ability to find its own structure. The linking of amino acids into chains is therefore more than simple construction. It sets the stage for everything that follows. The sequence determines the range of possible shapes, while the environment influences which of those shapes will be favored. Together, they create a system that is both flexible and reliable. As rest continues and awareness softens, it can be comforting to reflect on this steady molecular activity. Even in stillness, amino acids are linking into chains. Chains are beginning to bend, and proteins are quietly preparing to take on their roles. There is no urgency in this work, only continuity. This linking process has been repeated countless times across the history of life. It is ancient, dependable, and deeply ingrained in the fabric of living systems. From the simplest organisms to the most complex, the same method is used, the same bonds formed, the same calm progression followed. By understanding how amino acids link into chains, we gain a deeper appreciation for the simplicity underlying biological complexity. Each connection is small and unremarkable on its own, yet together they give rise to structures capable of remarkable precision and subtlety. It is a reminder that life builds itself patiently, one bond at a time, guided by principles that favor balance and stability. As this part comes to a close, the growing chain remains flexible and open to possibility. Its final form has not yet emerged, but the groundwork has been laid. In the quiet environment of the cell, surrounded by water and time, the chain is ready to continue its journey toward folding, guided by the same gentle forces that have shaped life for billions of years. 5. The idea of folding. Once a protein exists as a long chain of linked amino acids, something subtle and almost inevitable begins to occur. The chain does not remain stretched out for long. Given time, warmth, and the presence of water, it begins to bend, curve, and settle. This gradual transformation is known as folding, and it is one of the most natural processes in molecular biology. Nothing pushes the chain into shape. Nothing instructs it step by step. Folding arises simply because the chain exists in an environment where certain arrangements feel more stable than others. To think of folding as an active task can be misleading. Proteins do not work to fold themselves, nor do they search for a correct form in any conscious way. Instead, folding is a process of release. The chain lets go of less stable arrangements and drifts toward ones that feel more comfortable at the molecular level. Much like a body shifting during sleep to find a more restful position, the protein moves toward balance without effort. At the heart of folding is the relationship between the protein and its surroundings. The interior of the cell is filled with water, and water plays a quiet but decisive role in shaping proteins. Some amino acid side chains interact easily with water, while others avoid it. As the chain moves, these tendencies begin to influence its shape. Parts that are comfortable near water remain exposed, while parts that are less compatible gradually turn inward. This simple preference alone drives much of the folding process. As the chain bends, certain regions may come close enough to form temporary contacts. These interactions are gentle and reversible. If a particular contact does not contribute to stability, it fades and the chain moves on. If it does reduce overall tension, it remains. Over time, these small decisions accumulate, guiding the protein toward a shape that balances all of its internal preferences. Folding does not happen all at once. It unfolds, quietly, in stages. The chain may briefly adopt one shape, then relax into another. Portions of the protein may settle early, while others remain flexible for longer. This gradual progression allows errors to correct themselves naturally. There is no rush to arrive at the final form. One of the most calming aspects of protein folding is how predictable it is. Given the same sequence of amino acids and the same environment, a protein will fold into the same shape again and again. This reliability allows cells to function smoothly, producing proteins with confidence that they will behave as expected. Folding is not a gamble. It is a well-worn path shaped by physical law. It is worth pausing on the idea that folding is guided by physics rather than biology in the narrow sense. The forces involved are the same ones that govern all matter: attraction and repulsion, movement toward lower energy states, and the tendency of systems to settle into balance. Biology does not override these principles, it relies on them. Life is built on the quiet predictability of physical interactions. When a protein folds, it moves toward a state of lower free energy. This does not mean it becomes rigid or immobile. Rather, it finds a shape where the overall system is more stable than before. In this state, the protein can perform its function efficiently, interacting with other molecules in a controlled and repeatable way. Folding, then, is not an end point, but a preparation. The folded protein remains dynamic. It may breathe slightly, shifting its structure in small ways as it interacts with its environment. These movements are subtle and constant, allowing the protein to respond to changes without losing its overall form. Stability and flexibility coexist, reinforcing one another. The idea of folding also highlights an important distinction. A protein sequence determines what shapes are possible, but the environment influences which shape is realized. Temperature, chemical conditions, and the presence of other molecules all play a role. Under normal cellular conditions, folding proceeds smoothly. When conditions change, folding may slow or take alternative paths. Even then, the system remains adaptable. Within the crowded cell, folding often begins while the protein is still being assembled. As the chain emerges, early segments may start to bend and align, setting the stage for later folding steps. This overlapping of creation and folding helps proteins reach their final forms efficiently without unnecessary wandering. Sometimes, folding benefits from gentle assistance. Other proteins may temporarily interact with the folding chain, preventing unwanted contacts or shielding sensitive regions. These helpers do not impose a shape. They simply create conditions that allow folding to proceed without interference. Once their task is done, they step away, leaving the protein to settle fully on its own. What is striking is how rarely folding fails under normal conditions. Given the complexity of proteins and the density of the cellular environment, one might expect frequent problems. Instead, folding is remarkably robust. This reliability speaks to the elegance of the underlying principles. Simple tendencies, applied consistently, produce dependable outcomes. From a broader perspective, folding reflects a theme that appears throughout nature. Systems tend to move toward states that require less energy to maintain. Rivers follow paths of least resistance. Objects fall into stable positions. Proteins fold for the same reason. They are not seeking purpose or function. They are simply responding to the conditions around them. In the context of rest and sleep, there is something reassuring about this idea. Even as conscious thought slows, even as the body relaxes, countless proteins are folding quietly, maintaining the structures and processes that support life. This work does not demand attention. It continues automatically, guided by the same gentle rules that allow rest itself to occur. Folding also reminds us that complexity does not always require control. Order can emerge without oversight. Structure can arise from interaction rather than instruction. In the cell, this principle allows life to sustain itself efficiently, without constant correction or intervention. As this part draws to a close, the protein chain has begun its transformation. It is no longer just a sequence of linked amino acids. It is becoming a shape, a form suited to a particular role. This transformation is not dramatic or abrupt. It is gradual, calm, and deeply reliable. In the quiet interior of the cell, folding continues, moment by moment, protein by protein. Each one follows its own path toward balance, guided by preferences encoded in its sequence and shaped by its surroundings. Together, these countless folding events create the steady foundation upon which all living activity rests, unfolding gently, even as rest deepens and awareness drifts further into calm. 6. Why shape matters more than size. As a protein continues to fold, its growing shape begins to define what it can and cannot do. In molecular biology, function does not arise from how large a molecule is or how much space it occupies, but from the precise way it is shaped. A protein's size may be modest or substantial, yet without the correct contours, its presence would be meaningless. Shape is what allows a protein to interact, to recognize, and to participate in the quiet choreography of cellular life. At this scale, shape is not an abstract idea. It is physical and specific, defined by curves, pockets, ridges, and surfaces. These features determine how a protein fits with other molecules, much like how two pieces of a puzzle align. When the shape is right, interaction occurs naturally. When it is not, the molecules simply pass by one another without effect. There is no struggle, no forceful attempt to connect. Compatibility is enough. This focus on shape explains why folding is so essential. A protein that remains unfolded or incorrectly folded may still exist as a chain of amino acids, but it lacks identity. Without its proper form, it cannot participate meaningfully in cellular processes. Folding transforms a generic chain into a specific tool, one capable of performing a defined role again and again. Many proteins act by binding to other molecules. Enzymes, for example, guide chemical reactions by holding their substrates in just the right position. This guidance does not come from strength, but from fit. The enzyme's shape creates a small environment where a reaction becomes easier to complete. The protein does not force the reaction to happen. It simply makes the preferred outcome more likely. Other proteins serve as messengers or receptors, recognizing specific signals and responding accordingly. Their shapes allow them to distinguish one molecule from another, even among many similar options. This selectivity depends on subtle differences in form, not on mass or volume. A slight change in shape can alter a protein's behavior completely. Structural proteins provide another perspective on the importance of shape. These proteins form fibers, sheets, or networks that support cells and tissues. Their shapes allow them to connect to one another in repeating patterns, creating strength and resilience. Again, it is the arrangement of the molecule, not its size, that determines its contribution. What is especially calming about this reliance on shape is its precision. Proteins do not act broadly or indiscriminately. They engage only with what they are suited to interact with. This specificity reduces unnecessary activity and helps maintain balance within the cell. Each protein occupies a niche defined by its structure, fitting naturally into the larger system. The folding process ensures that this structure emerges reliably. As the protein settles into its lowest energy state, its shape becomes stable enough to persist, yet flexible enough to adapt slightly when needed. This balance allows proteins to function under a range of conditions without losing their identity. It is also important to recognize that shape is not a static feature. Proteins may shift subtly as they work, opening and closing small regions, adjusting their surfaces as interactions occur. These movements are part of their function, not signs of instability. The folded structure provides a framework within which motion can occur safely and predictably. The relationship between shape and function also explains why small changes in sequence can have significant effects. Altering even a single amino acid can change how a protein folds, and thus how it behaves. Sometimes these changes are minimal, barely noticeable. Other times, they prevent the protein from functioning at all. This sensitivity highlights the care with which folding must proceed, yet also underscores the reliability of the process when conditions are normal. From a broader view, the emphasis on shape over size reflects a recurring principle in nature. Efficiency often arises from form rather than magnitude. Small structures, arranged thoughtfully, can produce powerful effects. Proteins embody this principle at the molecular level, achieving remarkable outcomes through precise folding. As the body rests, countless proteins are carrying out their roles based on their shapes alone. They bind, release, catalyze, support, and signal, all without awareness or intention. Their success depends on having folded correctly, finding the form that allows them to interact smoothly with their surroundings. There is something deeply reassuring in this quiet dependence on shape. It suggests that life is sustained not by constant exertion, but by structures that naturally fit together. When proteins fold properly, they do not need supervision. They simply do what they are shaped to do. In this way, folding can be seen as an act of preparation rather than completion. The protein does not achieve its purpose by folding. Folding merely makes purpose possible. Once shaped, the protein enters the ongoing flow of cellular life, interacting, adjusting, and contributing as needed. As this part comes to a gentle close, the importance of folding becomes clearer. Shape is the language proteins use to communicate and cooperate. Without it, the molecular world would be silent and uncoordinated. With it, life proceeds smoothly, guided by countless small interactions that require no attention and no effort. In the quiet interior of the cell, proteins continue to settle into their forms, finding shapes that allow them to serve. These shapes persist through rest and activity alike, supporting life with steady reliability. And so, as awareness drifts and rest deepens. This molecular precision continues undisturbed, shaping function from form, moment by moment, without sound or strain. 7. Secondary structure. Repeating patterns. As a protein continues its gentle journey from a flexible chain toward a stable form, certain patterns begin to appear along its length. These patterns are not imposed from outside, nor are they planned in advance. They arise naturally from the structure of the protein's backbone and the quiet tendencies of its chemical bonds. This stage of folding is known as secondary structure, and it represents one of the most soothing ideas in molecular biology: repetition as a source of stability. The backbone of a protein, formed by the repeating sequence of peptide bonds, has a regularity that invites order. Along this backbone are atoms capable of forming weak attractions with one another, particularly hydrogen bonds. These bonds are not strong or rigid, but they are consistent. When the chain bends in certain ways, these hydrogen bonds align easily, reinforcing the shape that brought them together. Over time, these reinforced shapes persist, becoming recognizable patterns. Two such patterns appear again and again across nearly all proteins. One is a gentle spiral known as the alpha helix. In this form, the protein backbone coils smoothly around an invisible axis, with each turn supported by hydrogen bonds linking nearby sections of the chain. The result is a structure that is compact, flexible, and resilient. It resembles a softly wound spring, able to compress slightly and then return to its original shape. The other common pattern is the beta sheet. Here, sections of the protein chain stretch out and lie alongside one another, forming a sheet-like arrangement. Hydrogen bonds connect neighboring strands, holding them together in a stable, flat structure. These sheets may run in the same direction or in opposite directions, but in either case, the result is a calm, ordered surface that adds strength and definition to the protein. What makes these patterns especially reassuring is their predictability. Given the same sequence and environment, alpha helices and beta sheets form reliably. They do not require supervision or correction. The protein backbone naturally favors these arrangements because they allow hydrogen bonds to form efficiently, lowering the overall energy of the system. In this way, repetition becomes a path to rest. Secondary structures often appear early in the folding process. As soon as the chain is long enough, certain regions may begin to coil or align, even before the entire protein has been assembled. These early patterns provide a framework for later folding, guiding the protein toward its final shape without locking it into place too soon. It is important to note that not every part of a protein adopts one of these repeating patterns. Between helices and sheets are flexible regions known as loops or turns. These segments lack regular hydrogen bonding and remain more mobile. Far from being flaws, these flexible regions are essential. They allow the protein to connect structured elements smoothly and to adjust its shape when interacting with other molecules. The balance between repetition and flexibility is central to protein function. Secondary structures provide stability, while unstructured regions provide adaptability. Together, they create a molecule that is both reliable and responsive, capable of maintaining its form while engaging with a changing environment. The presence of these repeating patterns also helps explain how proteins can fold efficiently despite their size. Rather than attempting to settle all at once into a complex three-dimensional form, the protein builds itself gradually. Local patterns form first, stabilizing small regions. These regions then interact with one another, guiding the protein toward its overall structure. Folding becomes a sequence of manageable steps rather than a single, overwhelming transition. Secondary structure is determined primarily by interactions along the backbone, rather than by the specific side chains of amino acids. This means that many different sequences can form similar helices or sheets. The individuality of a protein emerges later, when these elements are arranged relative to one another. At this stage, the focus is on establishing a calm internal order. There is something deeply comforting about the universality of these patterns. Alpha helices and beta sheets are found in proteins from all forms of life, from the simplest organisms to the most complex. This shared structural language suggests a continuity that stretches across evolutionary time, linking all living systems through common molecular principles. As proteins fold within the cell, these patterns appear quietly, without disturbance. The surrounding environment supports them, maintaining conditions where hydrogen bonds can form and persist. Water molecules, temperature, and chemical balance all contribute to this stability, allowing secondary structures to emerge naturally. These repeating forms also lend themselves to cooperation. Helices may pack together, sheets may stack or curve, and combinations of both can create intricate yet stable assemblies. Each secondary structure retains its identity while contributing to a larger whole. This layered organization mirrors the way life itself is built, from simple units arranged into complex systems. While secondary structures are stable, they are not rigid. They can bend slightly, shift, or unwind under certain conditions, then reform when balance is restored. This resilience ensures that proteins can withstand minor disturbances without losing their function. Stability here does not mean immobility, but rather a tendency to return to form. As rest deepens and attention softens, it may be soothing to reflect on these quiet patterns forming within. Even now, proteins are coiling into helices and aligning into sheets, establishing the foundations of their final shapes. This work proceeds without effort, guided by repetition and gentle attraction. Secondary structure demonstrates that order does not require complexity at every level. Simple patterns, repeated consistently, can provide a strong and reliable framework. From this framework, diversity and function can emerge. It is a reminder that calm repetition is not stagnation, but preparation. As this part draws to a close, the protein has acquired its first recognizable shapes. Spirals and sheets have formed, lending stability and direction to the folding process. These patterns will soon interact, combining into larger structures that define the protein's unique role. For now, they exist as quiet, repeating motifs, holding the chain in gentle balance. Within the cell, this process continues uninterrupted, moment by moment. Proteins fold not through force, but through preference, not through urgency, but through repetition. And so, as the body rests and the mind drifts, these molecular patterns persist, steady and untroubled, shaping life from within through their calm and reliable order. 8. Tertiary structure, settling into form. As secondary structures quietly establish themselves along the protein chain, a deeper level of organization begins to emerge. The spirals and sheets do not remain isolated from one another. Instead, they start to interact, guided by the same gentle tendencies that shaped them in the first place. This stage of folding is known as tertiary structure, and it represents the moment when a protein settles into a cohesive three-dimensional form that reflects its full identity. At this point, the protein is no longer simply a collection of local patterns. It becomes a unified whole. Regions that were once distant along the sequence may now find themselves close in space, drawn together by the combined preferences of their amino acid side chains. These interactions are subtle, yet decisive. They determine how the protein holds itself together and how it presents itself to the surrounding cellular environment. The forces involved in forming tertiary structure are varied, but none are abrupt or harsh. Some side chains are drawn toward one another because their electrical charges complement each other. Others cluster together because they share a mutual avoidance of water. Still others form brief, stabilizing attractions that help anchor different regions of the protein in place. Each interaction contributes a small amount of stability, and together they create a structure that is greater than the sum of its parts. What is especially calming about this process is its gradual nature. The protein does not snap into its final shape, it eases into it. As interactions form, the chain adjusts, releasing less stable arrangements and reinforcing those that feel more balanced. This process may involve many small shifts, each one moving the protein closer to a state where internal tensions are minimized. Tertiary structure reflects the protein's response to its environment as much as its internal composition. Water continues to play a central role, encouraging certain regions to remain on the surface while others fold inward. The result is often a protein with a compact interior, shielded from water, and a surface that interacts comfortably with its surroundings. This arrangement is not planned. It emerges naturally from the chemical properties of the amino acids involved. As the protein settles, it may briefly explore alternative shapes. These intermediate forms are part of the folding journey, not signs of error. They allow the protein to sample possibilities and discard those that are less stable. Over time, one configuration proves most favorable, and the protein remains there, gently fluctuating, but largely consistent. The concept of tertiary structure highlights an important principle in biology. Stability does not require rigidity. A folded protein is stable because it occupies a low-energy state, not because it is locked in place. It can move slightly, flexing as it interacts with other molecules, without losing its overall form. This balance allows proteins to function efficiently in a dynamic environment. Many proteins rely on their tertiary structure to create specific functional features. Small pockets may form on the surface, capable of binding other molecules. Grooves or channels may appear, guiding substances along defined paths. These features arise naturally from the way the protein folds, without any need for external shaping. Form follows from interaction. In some cases, parts of the protein that appear unstructured at earlier stages become more defined during tertiary folding. Loops may settle into consistent positions, stabilized by nearby interactions. Other regions may remain flexible, contributing to the protein's ability to adapt. The final structure reflects a compromise between stability and motion, allowing the protein to remain reliable while responsive. Within the cell, tertiary folding often completes shortly after the protein is fully assembled. The environment supports this completion, providing a steady temperature and chemical balance. When assistance is needed, helper proteins may remain nearby, ensuring that folding proceeds smoothly. Once the protein has settled, these helpers withdraw, leaving the protein to function independently. The remarkable thing about tertiary structure is how repeatable it is. Each time a protein with a given sequence is produced, it folds into essentially the same three-dimensional form. This consistency allows cells to rely on their molecular components, knowing that structure and function will align predictably. Folding becomes a dependable step in the ongoing rhythm of life. There is also an elegance to the way tertiary structure integrates earlier folding stages. Secondary structures are not discarded or overwritten. They are incorporated, arranged, and stabilized within the larger form. The protein's final shape honors its smaller patterns, weaving them together into a coherent whole. As rest deepens and awareness softens, it can be reassuring to imagine this quiet settling happening throughout the body. Proteins are finding their shapes, guided by gentle forces, supported by their environment. There is no rush, no urgency. Folding unfolds at its own pace, shaped by balance rather than demand. Tertiary structure represents a turning point in the life of a protein. Once this form is established, the protein is ready to engage fully with its surroundings. It can bind, catalyze, support, or signal, all because its shape now allows it to do so. Folding has transformed potential into capability, yet even now, the process remains reversible in subtle ways. If conditions change, parts of the protein may adjust, unfolding slightly and then refolding as balance is restored. This adaptability ensures resilience, allowing proteins to persist through minor fluctuations without losing function. As this part comes to a gentle close, the protein rests in its three-dimensional form, stable yet alive with small movements. Its tertiary structure holds it together, defining its role within the cell. Around it, countless other proteins are doing the same, each settling into form, each contributing to the quiet continuity of life. In the calm interior of the cell, this settling continues without interruption. Proteins fold, stabilize, and begin their work, all while the larger organism rests. It is a reminder that even in stillness, life remains active in the most peaceful way possible, sustained by structures that emerge naturally from simple, reliable principles. 9. The cellular environment. A protein never folds in isolation. From the moment it begins to take shape, it is surrounded by the living interior of the cell, an environment that is warm, fluid, and densely populated with other molecules. This setting is not chaotic, though it is busy. It is a carefully balanced space where countless processes unfold simultaneously, each supported by conditions that favor stability and gentle change. Understanding protein folding means understanding this environment, because the cell itself plays a quiet but essential role in guiding proteins toward their final forms. The interior of a cell is mostly water. This water is not empty or inert, but active and responsive, forming temporary associations with other molecules and influencing how they behave. For proteins, water is a constant presence, shaping folding by encouraging some regions to remain exposed while others turn inward. Amino acid side chains that interact comfortably with water tend to face outward, while those that do not are gently guided toward the protein's interior. This simple preference helps establish the overall structure without any need for instruction. Beyond water, the cell contains a wide variety of dissolved substances. Ions such as sodium, potassium, and calcium drift through the cytoplasm, contributing to its chemical balance. Small molecules, sugars, lipids, and metabolites move steadily from place to place. Other proteins are everywhere, some newly formed, others long established, all sharing the same limited space. Despite this crowding, folding proceeds smoothly, a testament to the adaptability of biological systems. This crowded environment actually supports folding rather than hindering it. The presence of other molecules limits the range of shapes a protein can adopt, gently steering it away from extended or unstable configurations. In this way, crowding acts as a subtle guide, encouraging compact and efficient forms. What might seem like an obstacle becomes a quiet aid. Temperature within the cell is another important factor. It is warm enough to allow molecular movement, yet stable enough to prevent excessive agitation. This balance ensures that proteins have the flexibility needed to fold, while maintaining the integrity of the interactions that stabilize their structures. Folding becomes a calm negotiation between motion and rest. The chemical conditions of the cell, including acidity and ion concentration, are carefully regulated. These conditions influence how amino acid side chains interact, affecting attractions and repulsions within the protein. Under normal circumstances, this balance remains steady, allowing proteins to fold predictably. When conditions shift, folding may adjust accordingly, demonstrating the responsiveness of the system. Within this environment, proteins often begin folding as they are being synthesized. As the chain emerges, early segments encounter water and nearby molecules, starting to bend and form patterns. This early interaction with the cellular environment helps guide folding efficiently, reducing the need for large-scale rearrangements later on. The cell also provides specialized regions where folding can occur under particularly favorable conditions. Certain compartments offer controlled environments that protect proteins as they fold, shielding them from unnecessary interactions. These spaces do not rush the process. They simply offer consistency and calm, allowing folding to proceed without interruption. It is important to note that the cell does not micromanage folding. It does not dictate each step or correct every movement. Instead, it maintains conditions that allow physical principles to operate reliably. The cell's role is one of support rather than control, trusting that proteins will find their forms given time and stability. Other molecules within the cell may briefly interact with folding proteins, not to reshape them, but to prevent unwanted contacts. These interactions are temporary and gentle, ensuring that proteins do not become tangled or stuck. Once folding is complete, these helpers move on, leaving the protein free to function. The cellular environment also plays a role in what happens after folding. Proteins that have settled into stable forms are integrated into the ongoing activity of the cell. They may remain where they are or be transported to other locations, all within the same supportive environment. Proteins that fail to fold properly are recognized and recycled. Their components return to the pool of available amino acids. This process occurs quietly, without alarm. There is a remarkable efficiency to this system. Proteins are folded, used, and eventually replaced, all within the same cellular space. Nothing is wasted, and nothing is rushed. The environment supports continuity, allowing life to persist through constant renewal. From a restful perspective, there is something deeply soothing about this picture. Even as the body rests, the cellular environment remains active in the most gentle way. Proteins fold, interact, and perform their roles without any need for conscious involvement. The same physical principles that allow a protein to settle into shape also allow the body to relax into rest. The cell's interior can be imagined as a softly lit room filled with quiet activity. Molecules drift, meet, separate, and meet again. Folding occurs not as a dramatic event, but as a natural consequence of being present in this space. Stability arises from consistency, not from effort. As this part comes to a close, it becomes clear that protein folding cannot be separated from its surroundings. The cellular environment is not merely a backdrop, but an active participant, shaping and supporting the process at every stage. Without this environment, folding would be unpredictable. With it, folding becomes one of the most reliable processes in biology. In the calm interior of each cell, proteins continue to fold and function, sustained by water, temperature, and balance. This quiet collaboration between molecule and environment unfolds continuously, even as awareness drifts and rest deepens. Life persists not through strain, but through the steady support of conditions that allow simple principles to operate smoothly, moment by moment, without interruption. 10 molecular attractions and preferences. Within the folded and folding protein, there is no single force at work, no dominant pull that dictates form. Instead, shape emerges from a collection of small attractions and gentle preferences, each contributing quietly to the whole. These molecular tendencies are modest on their own, yet together they guide proteins toward stable, functional forms. Understanding protein folding at this level is less about mechanics and more about harmony, about how many subtle influences align to produce balance. One of the most important of these influences is the attraction between opposite electrical charges. Some amino acid side chains carry a slight positive charge, while others carry a slight negative one. When these opposites come near one another within the folding protein, they are gently drawn together. This attraction is not forceful or abrupt. It is more like a quiet inclination, encouraging certain regions of the protein to remain close. These interactions help stabilize the overall structure, anchoring parts of the chain in place. Alongside these charged interactions are hydrogen bonds, which form between specific atoms along the protein backbone and side chains. Hydrogen bonds are individually weak, easily formed and broken, yet when many of them align, they provide remarkable stability. They are responsible for holding together alpha helices and beta sheets, and they continue to contribute to stability as these structures assemble into larger forms. Their strength lies not in rigidity, but in numbers and consistency. Another important influence arises from how amino acid side chains interact with water. Some side chains blend easily with the surrounding water, forming comfortable associations. Others do not. These water-avoiding side chains tend to cluster together, away from the watery environment of the cell. This tendency, often called the hydrophobic effect, is one of the most powerful drivers of protein folding. It encourages the protein to adopt a compact shape, with its water-avoiding regions tucked safely inside. This movement inward is not a response to pressure or command. It is simply the result of preference. Water molecules form more stable arrangements when hydrophobic side chains are removed from their presence. In response, the protein settles into a form that benefits both itself and its environment. Folding, in this sense, is a cooperative act between molecule and surroundings. There are also interactions known as van der Waals forces, which arise from tiny fluctuations in electrical charge. These forces are extremely weak and operate only at very close distances. Yet when many such contacts occur between tightly packed regions of a folded protein, they add to the overall stability. They help fine-tune how closely different parts of the protein fit together, smoothing the final form. What is remarkable is how these different attractions coexist without conflict. None of them dominates entirely. Instead, they balance one another, each influencing folding just enough to contribute to a stable outcome. If one interaction weakens, others often compensate. This redundancy ensures reliability, allowing proteins to fold correctly even in the presence of small disturbances. These molecular preferences also explain why folding is reversible under certain conditions. If temperature rises too high or chemical balance shifts significantly, some interactions weaken. Hydrogen bonds may break more easily, charged attractions may lessen, and hydrophobic clustering may loosen. When conditions return to normal, these preferences reassert themselves, and the protein may refold. This adaptability allows life to persist through change. It is important to recognize that none of these attractions requires energy input to function. Proteins do not consume energy to fold. Folding occurs because it leads to a state that is energetically favorable. The protein and its environment together find a configuration that requires less maintenance to sustain. In this way, folding aligns naturally with the principle of energy efficiency that governs all physical systems. As proteins fold, they continuously test these interactions. A region may form a charged contact, then release it if it proves unstable. A hydrophobic cluster may tighten or loosen as the chain adjusts. This ongoing evaluation happens silently, without error messages or corrections. The protein simply drifts toward arrangements that feel more balanced. From a broader perspective, these molecular attractions mirror processes found throughout nature. Sand settles into stable piles. Water flows into low places. Objects come to rest where forces are balanced. Protein folding follows the same logic, operating at a scale too small to see, yet governed by the same principles. In the quiet interior of the cell, these interactions occur constantly. Even now, proteins are adjusting their shapes slightly, responding to tiny changes in their surroundings. These movements are not signs of instability, but of responsiveness. A folded protein is alive with subtle motion, always poised to interact when needed. As rest deepens, there is something comforting in knowing that life is sustained by preferences rather than pressures. Proteins do not need to be forced into place. They simply settle where they belong, guided by attractions that favor balance and harmony. This process unfolds continuously, without strain. The reliability of protein folding depends on the consistency of these molecular preferences. Because the physical properties of amino acids and water remain stable, folding remains predictable. Cells can trust that proteins will assume their proper forms, allowing larger biological processes to proceed smoothly. As this part draws to a close, the protein stands as a testament to cooperation at the smallest scale. Charged attractions, hydrogen bonds, hydrophobic tendencies, and countless tiny forces have come together to shape it. None acted alone, and none needed direction. Together, they created a form that fits naturally into the living system. Within every cell, this quiet interplay continues. Proteins fold and refold, held together by preferences that favor stability and release what does not serve. Life persists through this gentle negotiation, unfolding moment by moment, even as the body rests and awareness drifts into calm. 11. Molecular chaperones, quiet assistants. Within the crowded interior of the cell, protein folding is remarkably reliable. Yet the environment is not entirely without challenges. Many molecules move through the same space, and newly forming protein chains can sometimes encounter distractions or obstacles along their path toward stability. To support folding without disrupting its natural rhythm, Cells make use of a special group of proteins known as molecular chaperones. These helpers do not instruct, reshape, or force. Instead, they offer quiet assistance, creating conditions in which folding can proceed calmly and correctly. The name chaperone may suggest supervision or control, but at the molecular level, the role is far gentler. Chaperone proteins do not determine a protein's final shape. They do not carry blueprints or impose structure. The information needed for folding still resides entirely within the amino acid sequence of the protein itself. Chaperones simply help ensure that this information can be expressed without interference. As a new protein chain emerges into the cellular environment, it is flexible and open to many possible interactions. Some of these interactions are helpful, guiding folding forward. Others are less useful, drawing the chain into temporary tangles or contacts that slow progress. In such moments, chaperones may bind briefly to exposed regions of the chain, shielding them from unwanted encounters. This binding is not permanent. Once the risk has passed, the chaperone releases the protein, allowing folding to continue. What is striking about this assistance is how restrained it is. Chaperones do not cling or dominate. Their interactions are reversible and precisely timed. They step in when needed and step away when their presence is no longer beneficial. This restraint ensures that the protein retains freedom to explore its own folding path. Some chaperones provide enclosed spaces where folding can occur in relative isolation. These structures offer a calm environment, temporarily separating the folding protein from the surrounding crowd. Inside this space, the protein can bend and adjust without distraction. When folding is sufficiently complete, the protein is released back into the cell, ready to take on its role. Even in these enclosed settings, chaperones do not dictate outcomes. They do not guide the protein toward a specific shape. They merely prevent premature or unhelpful interactions, trusting the protein's own tendencies to lead it toward balance. Folding remains a self-organizing process, supported rather than directed. The presence of chaperones highlights an important principle in biology. Support does not require control. Systems can be assisted without being overridden. By reducing noise and interference, chaperones allow natural processes to unfold more smoothly. This approach is both efficient and gentle, minimizing the need for correction later on. Chaperones are especially important during times of increased activity or stress within the cell, when many proteins are being produced at once, or when conditions fluctuate slightly. The risk of misfolding can increase. In these moments, chaperones quietly increase their activity, providing additional support without altering the fundamental process. Once conditions return to normal, their involvement naturally diminishes. This adaptability reflects the broader resilience of cellular systems. Rather than preventing all variation, cells tolerate small fluctuations and respond as needed. Chaperones are part of this responsive network, stepping in when balance is threatened and stepping back when it is restored. It is also worth noting that chaperones themselves are proteins, folded and maintained by the same principles they support. They are part of the same molecular community, subject to the same rules of attraction and stability. Their ability to assist others arises from their own carefully folded structures, which allow them to bind and release in controlled ways. From a restful perspective, there is something reassuring about the presence of these quiet helpers. They do not rush or panic when folding encounters difficulty. They respond calmly, providing space and time rather than force. This approach mirrors the way healthy systems at all scales maintain balance, favoring gentle correction over drastic intervention. As proteins fold with chaperone assistance, they retain full ownership of their final forms. The role of the chaperone ends once stability is achieved. There is no lingering dependence. The protein moves forward independently, integrating into the ongoing activity of the cell. This temporary support model ensures efficiency. Chaperones are free to assist many proteins over time, moving from one task to another as needed. They do not become permanent fixtures, but remain available, adaptable, and responsive. In the quiet interior of the cell, these interactions unfold without awareness or effort. chaperones bind and release, proteins fold and settle, and life continues smoothly. This constant, yet gentle activity supports the larger rhythms of the organism, allowing rest, movement, and renewal to occur without disruption. As awareness softens and rest deepens, it can be comforting to know that even at the smallest scales, systems exist to provide calm assistance when needed. Protein folding is not left entirely to chance, nor is it rigidly controlled. It is supported by a network of quiet helpers that value balance over force. As this part comes to a gentle close, the image that remains is one of cooperation without command. Molecular chaperones stand nearby, attentive but unobtrusive, ready to help when folding needs a little space. Through their presence, proteins are allowed to become what they are meant to be, guided by their own structure and supported by an environment that favors calm and stability. Within every cell, this quiet assistance continues, unseen and uncelebrated, sustaining life through patience and restraint. Even as the body rests, these helpers remain at work, ensuring that the delicate process of folding proceeds smoothly, moment by moment, without strain or disturbance. 12. Energy landscapes and stability. As proteins fold and settle into form, their movement can be understood through a quiet and deeply calming idea known as the energy landscape. This concept does not describe a physical place, but a way of imagining how a protein naturally drifts towards stability. Rather than wandering aimlessly or choosing randomly among shapes, the protein follows gentle slopes of decreasing energy, guided by balance rather than direction. At every moment, a protein exists in a particular energetic state. Some arrangements of its atoms require more energy to maintain, while others require less. High energy states are unstable and fleeting, while low energy states are comfortable and persistent. Folding is simply the process by which the protein moves from less stable arrangements toward those that are more stable, much like a ball rolling downhill until it comes to rest in a shallow valley. This movement does not happen all at once. The energy landscape is not a single smooth slope, but a broad surface with many small dips and rises. As the protein folds, it may pause briefly in intermediate states, settling momentarily before continuing onward. These pauses are not mistakes or failures. They are natural resting points along the path toward greater stability. The idea of an energy landscape helps explain why folding is both reliable and flexible. While there may be many possible routes toward the final form, most of them lead downward, toward lower energy. This means that even if the protein takes a slightly different path each time it folds, it is still guided toward the same stable outcome. Variability in motion does not undermine consistency in result. At the lowest point of this landscape lies the protein's native state, the folded form in which it functions most effectively. This state represents a balance of all molecular preferences acting together. Charged interactions, hydrogen bonds, hydrophobic clustering, and countless tiny forces all find their place here. No single interaction dominates. Instead, stability emerges from cooperation. It is important to understand that the native state is not the absolute lowest energy state imaginable, but the lowest accessible state under normal cellular conditions. This distinction allows proteins to remain functional. If they were locked into an unchangeable minimum, they would be too rigid to interact. By occupying a stable yet accessible valley, proteins can function while remaining responsive. The energy landscape is shaped by the protein's amino acid sequence and by its environment. Changes in temperature, chemical balance, or surrounding molecules can gently reshape the landscape, making some paths steeper or shallower. When conditions return to normal, the landscape returns as well, and folding proceeds as before. This responsiveness allows proteins to adapt without losing identity. One of the most reassuring aspects of this process is that proteins do not need to search exhaustively through all possible shapes. The energy landscape naturally funnels them towards stability. As unstable arrangements are abandoned quickly, the protein spends more time exploring configurations that are already close to its final form. Folding becomes efficient not through speed, but through preference. This funnel-like quality explains how even large proteins can fold reliably. While the number of possible configurations is enormous, most of them are energetically unfavorable and therefore rarely visited. The protein's movement is constrained gently but effectively, guided by the shape of the landscape itself. Stability, in this context, does not mean immobility. Even in its native state, a protein continues to fluctuate slightly, responding to thermal motion and interactions. These movements occur within the valley of the energy landscape, not outside it. The protein remains stable overall, even as it breathes gently at the molecular level. The concept of energy landscapes also helps explain why folding can recover after disruption. If a protein unfolds partially due to a temporary change in conditions, it does not lose its way. Once conditions stabilize, the landscape reasserts itself, and the protein naturally drifts back toward its native state. Folding does not need to be restarted from the beginning. It resumes from wherever the protein finds itself. Within the cell, this reliability is invaluable. Proteins can endure small fluctuations without permanent damage, contributing to the resilience of biological systems. Life does not depend on constant perfection, but on the ability to return to balance. As rest deepens, it can be soothing to imagine these energy landscapes at work throughout the body. Countless proteins are rolling gently downhill, settling into forms that support life without effort or awareness. Their motion is slow, steady, and guided by principles older than life itself. The idea of an energy landscape also reinforces the sense that folding is not a task to be completed, but a state to be maintained. Proteins do not finish folding and then stop. They remain within their stable valleys, adjusting subtly as needed, always ready to respond. This perspective aligns beautifully with the rhythms of rest and sleep. Just as the body settles into a comfortable position and remains there with small adjustments, proteins settle into stable forms and remain poised for activity. Stability here is not rigid stillness, but calm readiness. As this part comes to a gentle close, the image that remains is one of natural ease. Folding is not driven by effort, instruction, or urgency. It is guided by landscapes shaped by physics and chemistry, inviting proteins toward balance. In these landscapes, stability is not imposed, but welcomed. Within every cell, these quiet journeys continue uninterrupted. Proteins move, settle, and remain, supported by the contours of their energy landscapes. Even as awareness drifts and the body rests, this molecular calm persists, sustaining life through gentle motion and enduring balance. 13. Folding is not instant. Protein folding is often imagined as a quick event, a moment where a chain suddenly snaps into place and becomes complete. In reality, folding is a gradual and patient process, unfolding over time through many small adjustments. There is no single instant where folding finishes. Instead, there is a steady progression, a quiet settling that reflects the calm logic of molecular systems. From the moment a protein chain begins to emerge, it starts responding to its surroundings. Early segments may form temporary structures, then relax again. Later regions may take longer to settle, exploring different arrangements before finding stability. Folding is not a straight line from beginning to end, but a gentle wandering through possibilities, guided by balance rather than speed. This gradual nature is not a weakness, it is a strength. By moving slowly and incrementally, proteins reduce the risk of becoming trapped in unstable configurations. Small adjustments allow the chain to correct itself naturally, releasing arrangements that do not contribute to overall stability. There is time to explore, time to adjust, and time to settle. Within the energy landscape, folding proceeds downhill overall, but the path includes small plateaus and shallow valleys. These intermediate states are part of the journey. The protein may linger briefly in one of these states, held by partial interactions that offer some stability. Eventually, a more favorable arrangement appears, and the protein continues its descent. These intermediate forms are not errors or failures. They are natural expressions of the protein's flexibility. Each one represents a compromise between competing interactions, resolved gradually as more of the chain becomes involved. Folding is a process of negotiation, where preferences are weighed and balanced over time. The pace of folding is influenced by many factors. Protein size matters, with longer chains generally taking more time to explore their possibilities. The cellular environment also plays a role, as temperature, chemical balance, and molecular crowding affect how quickly adjustments can occur. Yet even under ideal conditions, folding remains unhurried. There is no advantage to rushing. This patience ensures reliability. A protein that folds too quickly might settle into a shape that is locally stable but not optimal. By allowing time for rearrangement, the protein increases its chances of reaching the most balanced and functional form. Folding favors accuracy over speed. The gradual nature of folding also explains why assistance from chaperones is sometimes helpful. When a protein pauses in an intermediate state, chaperones may prevent unwanted interactions that could lock it into an unproductive shape. Once the protein is ready to continue, these helpers step away, allowing folding to resume naturally. It is important to recognize that even after a protein reaches its native state, folding is not entirely complete in a strict sense. The protein continues to fluctuate subtly, responding to thermal motion and interactions. These movements are small and contained, but they are ongoing. Folding, then, is not a finished task, but a maintained condition. This perspective aligns with the rhythms of living systems. Life rarely operates through sudden completions. Growth, repair, and adaptation all occur gradually, through continuous adjustment. Protein folding follows the same pattern, contributing to the steady resilience of cells. From a restful standpoint, there is comfort in this slowness. Folding does not demand urgency. It unfolds at its own pace, guided by gentle forces that do not tire or rush. Even as awareness drifts and the body relaxes, proteins continue their quiet adjustments, supported by time and stability. The idea that folding is not instant also helps explain how proteins can adapt to change. If conditions shift slightly, the protein may adjust its structure gradually, finding a new balance without losing function. When conditions return to normal, the protein drifts back toward its original form. This adaptability depends on folding being a flexible, ongoing process. Within the cell, countless proteins are at different stages of folding at any given moment. Some are just beginning, others are nearing stability, and still others are maintaining their folded forms. This continuous activity creates a dynamic yet calm molecular landscape where movement and rest coexist. As this part comes to a gentle close, the image that remains is one of patience. Protein folding is not a race to completion, but a slow settling into balance. Each adjustment is small, each movement guided by preference rather than force. In the quiet interior of the cell, folding continues without interruption. Proteins bend, adjust, and settle, given the time they need to find stability. Even as the body rests and the mind softens, this molecular patience persists, sustaining life through gradual change and enduring calm. 14. Avoiding crowding and confusion. Within the cell, space is limited and precious. Thousands of different molecules occupy the same interior at once, all moving gently, all carrying out their own roles. Proteins fold in the midst of this activity, not in isolation, but surrounded by potential partners, obstacles, and distractions. Yet despite this density, folding proceeds with remarkable calm and reliability. This is possible because the cell has evolved subtle ways of avoiding crowding and confusion, allowing proteins the room they need to become themselves. The cellular interior is often described as crowded, but this crowding is not disorderly. Molecules are in constant motion, yet their movements are constrained by balance and scale. Proteins do not collide violently or compete aggressively for space. Instead, they drift past one another, influenced by water, temperature, and gentle physical boundaries. Within this steady flow, folding unfolds naturally. One of the simplest ways cells reduce confusion is through timing. Proteins are not all produced at once. Their synthesis is spread out over time, ensuring that the environment does not become overwhelmed by too many unfinished chains at the same moment. This pacing gives each protein a better chance to fold without interference, reducing the likelihood of accidental entanglements. Location also matters. Some proteins begin folding in specific regions of the cell where conditions are especially favorable. These areas offer relative calm, shielding folding chains from unnecessary contact with other molecules. The protein does not need to be isolated completely. It only needs enough space and consistency to explore its folding options without being pulled in too many directions. As folding progresses, the protein naturally becomes more compact. This compactness itself helps reduce confusion. A folded or partially folded protein occupies less space and presents fewer exposed surfaces that could interact in unwanted ways. In this sense, folding helps protect itself, gradually limiting the number of possible distractions. Cells also make use of gentle separation. Certain proteins are directed to specific compartments, each with its own chemical character. These compartments differ slightly in composition, creating environments where particular proteins fold more easily. By distributing activity across spaces, the cell avoids overcrowding any single area. Another important factor is the reversible nature of most molecular interactions. When proteins come into brief contact, those interactions usually dissolve quickly unless they are specifically favored. This reversibility prevents accidental associations from becoming permanent problems. If a folding protein brushes against another molecule and the interaction is not helpful, it simply fades, and both molecules move on. Water continues to play a central role here. By surrounding proteins and filling the spaces between them, water acts as a buffer, softening encounters and discouraging prolonged, non-specific interactions. This buffering effect allows proteins to share space without becoming entangled, maintaining clarity within the crowded environment. The cell's internal organization also contributes to order. Although there are no rigid walls separating most molecules, patterns of movement and distribution emerge naturally. Larger structures tend to remain in certain regions, while smaller molecules move more freely. This self-organization reduces random encounters and creates gentle pathways through which folding proteins can move. When additional help is needed, chaperone proteins once again provide quiet support. They may temporarily bind to exposed regions of a folding protein, preventing it from sticking to the wrong partner. These interactions are brief and purposeful, designed only to reduce confusion. Once the risk has passed, the chaperone releases the protein, restoring its freedom. What is especially calming about these mechanisms is their subtlety. The cell does not impose strict separation or enforce rigid rules. Instead, it relies on tendencies, preferences, and timing. Folding is supported not through control, but through conditions that make the correct outcome more likely. This approach mirrors broader patterns in nature. Forests manage dense populations of organisms through spacing, timing, and balance. Rivers guide countless particles without collisions through flow and structure. At the molecular level, the cell achieves the same harmony, allowing many processes to coexist peacefully. As proteins fold successfully, they begin to contribute to this harmony themselves. Their folded forms are specific and selective, interacting only with appropriate partners. This specificity further reduces confusion, as proteins recognize what fits and ignore what does not. Folding thus strengthens order within the system. Even proteins that are no longer needed are handled calmly. They are broken down and recycled in a controlled manner, preventing accumulation that could disrupt folding elsewhere. This ongoing maintenance ensures that the cellular environment remains supportive rather than cluttered. From a restful perspective, there is reassurance in knowing that life manages complexity without strain. Even in crowded spaces, systems can remain clear and functional. Folding proteins are not overwhelmed by their surroundings. They are supported by an environment that favors patience and balance. As awareness softens, it can be soothing to imagine this quiet organization at work. Proteins fold, move, and settle without interference, guided by gentle boundaries and natural spacing. There is no rush, no competition, only a steady flow toward clarity. As this part draws to a close, the image that remains is one of calm coexistence. Within the cell, many molecules share the same space, yet confusion is rare. Folding proceeds smoothly because the environment itself encourages order through timing, spacing, and gentle support. Even as the body rests and the mind drifts, this molecular clarity persists. Proteins continue to find their forms, untroubled by the crowd around them. Life unfolds not through isolation, but through harmony, maintained quietly at the smallest scales, moment by moment, without effort or disturbance. 15. When folding takes a different path, even within the calm reliability of protein folding, there are moments when the process follows a slightly different course. These moments are not dramatic, nor are they catastrophic. They are simply variations that arise naturally in a complex, living environment. Folding does not always proceed along the smoothest possible route, and this flexibility is part of what allows life to persist in a world that is never perfectly uniform. As a protein moves through its energy landscape, it may sometimes settle temporarily into a shape that is not its final destination. These alternative arrangements can be stable enough to persist for a while, held in place by partial interactions that offer some balance. In most cases, the protein eventually continues onward, releasing these arrangements and finding its way to a more stable form. This wandering is not a failure, but an expression of the protein's freedom to explore. Occasionally, however, a protein may become more firmly held in one of these alternative shapes. This can happen if the environment shifts slightly, or if interactions form in an unexpected order. The protein is still folded in a sense, but its shape may not be ideally suited to its intended role. Even in these cases, the system responds calmly rather than urgently. Cells are well prepared for such variations. They do not assume that every folding event will be identical, nor do they demand absolute precision at all times. Instead, they rely on monitoring and gentle correction. Proteins that are not quite right are given opportunities to adjust, often with the quiet assistance of chaperones or changes in local conditions. It is important to recognize that folding along a different path does not necessarily mean harm. Many proteins are capable of functioning in more than one shape, adjusting their structures slightly in response to different tasks or environments. This adaptability allows proteins to participate in a range of interactions, increasing the versatility of cellular systems. Only when a protein's shape interferes with its ability to function or interact appropriately does the cell intervene more directly. Even then, the response is measured. The protein may be guided toward refolding, or it may be gently dismantled and recycled. This recycling is not wasteful or punitive. It is simply a way of maintaining balance, ensuring that resources are used effectively. The possibility of alternative folding paths highlights the importance of the environment once again. Small changes in temperature, chemical balance, or molecular crowding can influence which interactions form first, nudging the protein along one route or another. Because these conditions fluctuate naturally, variation in folding paths is expected rather than exceptional. What remains consistent is the system's ability to return to balance. When conditions stabilize, proteins are more likely to settle into their preferred forms. Folding remains guided by the same underlying preferences, even if the journey takes a slightly different route. From a broader perspective, this flexibility mirrors patterns found throughout biology. Development, growth, and adaptation rarely follow a single rigid plan. Instead, they allow for variation, correction, and adjustment. Protein folding reflects this same philosophy, favoring resilience over rigidity. As rest deepens, there is something reassuring in knowing that life does not depend on perfection at every step. Proteins are allowed to wander, to pause, and to adjust. Systems are in place to support them when they do. This tolerance allows life to continue smoothly, even in the face of constant small changes. Within the quiet interior of the cell, these alternative paths are explored without awareness or judgment. Folding continues, guided by balance rather than expectation. When a protein settles into a shape that serves, it remains. When it does not, the system responds gently, restoring equilibrium over time. As this part comes to a close, the image that remains is one of patience and flexibility. Protein folding is not a rigid script, but a calm improvisation guided by physical principles. Even when the path changes, the destination remains within reach. In the stillness of rest, this molecular adaptability continues unseen. Proteins fold, adjust, and refold as needed, supported by an environment that values balance over force. Life persists through this quiet willingness to allow variation, knowing that stability will return, moment by moment, without urgency or strain. 16. Cellular quality control. Within the calm and continuous activity of the cell, there exists a quiet system devoted to balance and maintenance. This system does not watch closely or intervene aggressively. Instead, it observes gently, responding only when necessary. It is often referred to as cellular quality control, yet it functions less like inspection and more like caretaking. Its purpose is simple: to ensure that proteins present in the cell remain useful, stable, and in harmony with their surroundings. Once a protein has folded, it is not immediately judged or tested. It begins its work naturally, interacting with other molecules as its shape allows. Most proteins perform their roles without difficulty, folding correctly and remaining stable for as long as they are needed. The cell does not interfere with these molecules. It trusts the folding process and allows proteins to function freely. Over time, however, conditions change. Proteins may experience gradual wear as they carry out repeated tasks. Small fluctuations in temperature or chemistry may affect their stability. Occasionally, a protein's shape may drift slightly away from its most balanced form. These changes are subtle and expected. The cell does not treat them as problems, but as part of the natural life cycle of molecules. Quality control within the cell is therefore patient and selective. It does not monitor every movement or adjustment. Instead, it responds when a protein's structure or behavior consistently deviates from what is useful. This response is not sudden or harsh. It unfolds in stages, offering opportunities for correction before removal is considered. One of the first steps in this gentle oversight is recognition. Certain molecular signals indicate whether a protein remains properly folded and functional. These signals are not alarms, but patterns of interaction. When a protein interacts smoothly with its environment, it is left undisturbed. When it begins to interact in unusual ways, attention is drawn quietly. In many cases, assistance is offered rather than removal. Chaperone proteins may return, binding briefly to help the protein regain a more stable shape. This process is calm and reversible. If the protein responds and settles back into balance, it continues its work as before. No record is kept, no penalty applied. The system simply moves on. When a protein no longer responds to gentle correction, the cell begins to prepare for recycling. This process is orderly and unhurried. The protein is guided towards specialized molecular machinery that carefully dismantles it into its basic components. Amino acids are released and returned to the cellular pool, ready to be used again. Nothing is wasted, and nothing is rushed. This recycling process serves several purposes at once. It prevents the accumulation of proteins that could interfere with other cellular activities. It recovers valuable building blocks, and it maintains the overall clarity of the cellular environment. All of this is achieved without disruption or urgency. What is especially calming about this system is its lack of judgment. Proteins are not labeled as good or bad. They are simply assessed in terms of usefulness and stability. When they no longer serve their role, they are gently returned to their basic form. This cycle of use and renewal supports the steady rhythm of cellular life. Quality control also adapts to changing conditions. During periods of increased stress or activity, the cell may temporarily enhance its monitoring, ensuring that folding and function remain reliable. When conditions normalize, this heightened attention fades. The system adjusts fluidly, without fixed thresholds or rigid rules. This adaptability reflects a broader principle seen throughout biology. Stability is maintained not through constant intervention, but through responsiveness. Systems remain calm by adjusting when needed and resting when they can. Protein quality control fits seamlessly into this pattern. From a restful perspective, there is reassurance in knowing that life is supported by systems that value balance over perfection. Proteins are not expected to last forever. They are expected to function for a time, then make way for new molecules. This acceptance of change allows the cell to remain flexible and resilient. As awareness softens, it may be comforting to imagine this quiet maintenance happening throughout the body. Proteins are monitored gently, assisted when needed, and recycled when their time has passed. This ongoing care requires no attention and no effort from consciousness. It unfolds automatically, guided by principles of balance and renewal. The presence of quality control does not undermine the reliability of protein folding. On the contrary, it enhances confidence in the system. Knowing that gentle correction and recycling are available allows folding to proceed without fear of error. Variation is tolerated because balance can always be restored. As this part comes to a gentle close, the image that remains is one of quiet stewardship. Within each cell, proteins are cared for rather than controlled. Folding is trusted, assistance is offered when needed, and renewal follows naturally. Life is sustained not by constant vigilance, but by calm responsiveness. Even as the body rests and the mind drifts, this molecular caretaking continues unseen. Proteins fold, function, and eventually give way to new ones, all within a system that values harmony and patience. Through this steady cycle, life maintains itself softly and continuously, moment by moment, without strain or disturbance. 17. Folding across all life. Protein folding is not a feature of one organism or one branch of life. It is a shared process, present wherever life exists. From the simplest, single-celled organisms drifting in water to the complex tissues of plants and animals, proteins fold according to the same quiet principles. This universality gives protein folding a special place in biology, linking all living things through a common molecular behavior that has persisted for billions of years. In the earliest forms of life, proteins already played essential roles. Even the most basic cells relied on folded proteins to carry out chemical reactions, maintain structure, and interact with their surroundings. The environment of early Earth was vastly different from today, yet the same physical laws applied. Water, temperature, and molecular attraction guided folding then, just as they do now. This continuity suggests that protein folding is not a fragile invention, but a deeply rooted feature of life itself. Across different species, the sequences of proteins may vary greatly, yet the folding process remains consistent. Amino acids link into chains, chains bend and settle, secondary structures form, and tertiary shapes emerge. The details differ, but the pattern endures. This shared process allows life to diversify while maintaining a stable molecular foundation. In bacteria, protein folding occurs in a small, densely packed interior where space is limited and efficiency is essential. Even here, folding proceeds smoothly, supported by chaperones and a carefully balanced environment. These organisms demonstrate that complex folding does not require elaborate structures or large spaces. Simple systems, when guided by reliable principles, can sustain life. In plants, protein folding supports processes as varied as photosynthesis, growth, and response to light. Proteins fold within cells that must also manage water balance, structural support, and energy capture. The same folding rules apply, allowing plants to build intricate systems from the same basic components used by all life. In animals, protein folding underlies movement, sensation, and thought. Muscles contract because proteins fold into shapes that allow interaction. Nerves transmit signals because folded proteins manage ion flow and communication. Even complex behaviors trace back to the quiet reliability of folding at the molecular level. What is especially calming about this universality is that folding does not become more complicated simply because life becomes more complex. The same physical tendencies apply at every scale. Life builds upward not by reinventing its foundations, but by reusing them in new combinations. This reuse allows for remarkable efficiency. Proteins that fold reliably in one organism can be adapted for use in another. Over evolutionary time, sequences change gradually, and folding patterns adjust accordingly. Yet the core process remains unchanged, offering stability amidst change. The shared nature of folding also reinforces the idea of biological unity. Despite the diversity of life forms, all rely on the same molecular choreography. Folding becomes a quiet language spoken by every cell, regardless of its origin or function. As rest deepens, there is something soothing in knowing that this process continues everywhere life exists. In oceans, soils, forests, and bodies, proteins are folding right now, guided by the same gentle preferences. Life persists not through constant novelty, but through faithful repetition of reliable processes. Even viruses, which depend on host cells to reproduce, rely on protein folding once their proteins are synthesized. Though they occupy a different place in biology, they too are subject to the same physical rules. Folding does not discriminate. It applies wherever proteins exist. This universality suggests that protein folding is not merely a biological curiosity, but a natural outcome of matter organized in certain ways. Given amino acids, water, and time, folding follows. Life emerges not through force, but through compatibility with physical law. As this part comes to a gentle close, the image that remains is one of quiet connection. Across all life, from the smallest to the most complex, proteins fold according to the same calm principles. This shared process binds living systems together, creating continuity across vast differences. Even as the body rests and awareness drifts, this molecular unity persists. Proteins fold in countless forms of life, sustaining activity and rest alike. Through this shared folding, life maintains itself softly and steadily, moment by moment, across the entire living world. 18. Self-organization in nature. As protein folding unfolds quietly within the cell, it offers a gentle illustration of a broader principle that appears throughout the natural world: self-organization. This principle describes how order can arise without external direction, how structure can emerge from interaction alone. Proteins do not require instructions to fold beyond the sequence they carry and the environment they inhabit. Given time and stable conditions, form arises naturally, guided by balance rather than command. Self-organization is not unique to biology. It can be observed wherever simple components interact repeatedly under consistent rules. Snowflakes form intricate patterns from water molecules responding to temperature and humidity. Sand arranges itself into ripples under the influence of wind and gravity. Rivers carve winding paths by following gradients in the landscape. In each case, no single element controls the outcome. Order emerges through cooperation. Protein folding follows this same logic. Amino acids link into chains. Chains encounter water in one another, and through countless small interactions, structure appears. No overseer dictates where a helix should form or when a sheet should align. These patterns arise because they are favored, because they reduce tension and increase stability. Folding is not a task performed by the protein, but a state it naturally approaches. What makes this idea especially calming is its lack of urgency. Self-organization does not hurry. It unfolds at a pace set by conditions rather than desire. Proteins fold as quickly or as slowly as the environment allows, guided by gentle preferences that never demand immediate resolution. This patience is built into the process itself. In the cell, self-organization allows complexity to exist without chaos. Thousands of proteins fold, interact, and function simultaneously, yet order persists. This is possible because interactions are local and reversible. Molecules respond to what is immediately around them, adjusting continuously. Large-scale order emerges from countless small, independent adjustments. Protein folding exemplifies this beautifully. Each interaction contributes a tiny piece of stability. No single bond defines the final structure. Instead, the protein settles into a form where all interactions are reasonably satisfied. Perfection is not required. Balance is enough. This approach allows proteins to remain flexible and responsive. Because folding is not locked into a rigid plan, proteins can adapt subtly to changes in their surroundings. When conditions shift, folding adjusts. When balance is restored, structure follows. Self-organization provides resilience through adaptability. The energy landscape concept fits naturally into this view. Proteins move through landscapes shaped by their own properties and their environment. They are guided downward toward stability, not by instruction, but by inclination. This movement is self-organizing, driven by the same principles that guide physical systems everywhere. As rest deepens, it can be soothing to reflect on how little effort is required for life to sustain itself at this level. Proteins fold without awareness, without intention, without strain. They do not need to know where they are going. The path emerges from interaction. This stands in contrast to many human-made systems, which often require constant control and adjustment. In nature, systems that self-organize are able to persist with minimal intervention. Protein folding is one such system, refined over evolutionary time to rely on physical law rather than oversight. Self-organization also explains how life could arise in the first place. Given the right components and conditions, molecules naturally formed more complex arrangements. Folding proteins would have emerged as stable structures long before cells became intricate. Life built itself gradually, guided by compatibility rather than design. Within modern cells, this same principle continues to operate. Folding is not an exception to order, but a foundation of it. Proteins find their places, interact selectively, and contribute to larger patterns of activity. From metabolism to movement, self-organized structures support every function. As awareness softens, it may be comforting to imagine this quiet order unfolding everywhere. Within each cell, countless self-organizing processes continue uninterrupted. Folding proteins are just one example of a world where complexity arises naturally, sustained by balance and repetition. As this part comes to a gentle close, the image that remains is one of trust in natural processes. Protein folding does not require control to succeed. It relies on the same principles that shape landscapes and weather patterns. Through self-organization, life maintains itself calmly and continuously. Even as the body rests and the mind drifts, this molecular order persists. Proteins fold, settle, and function without effort, guided by the quiet logic of nature. Through self-organization, stability emerges again and again, sustaining life in the most peaceful way possible. 19. The quiet elegance of molecular order. As we move closer to the end of this gentle exploration, it becomes possible to step back and observe protein folding not as a sequence of steps, but as a whole. When viewed from this wider perspective, folding reveals a quiet elegance that is easy to overlook when focusing only on details. It is an elegance rooted in order that arises naturally, without command or urgency, sustained by principles that are both simple and enduring. At the molecular level, order does not resemble the rigid arrangements often associated with precision. Instead, it is fluid and responsive, shaped by countless small interactions that align without force. Proteins fold because the conditions around them invite balance. Their shapes are not imposed, but welcomed. This distinction gives molecular order its calm character. Each folded protein represents a resolution of many competing tendencies. Some parts are drawn together, others held apart. Some regions remain flexible, others settle firmly into place. The final structure is not perfect in an abstract sense, but appropriate. It satisfies enough interactions to remain stable, while allowing movement where it is useful. This balance is the hallmark of molecular order. What makes this order especially elegant is its consistency. The same folding principles apply everywhere in the living world, regardless of scale or complexity. Whether a protein supports a simple chemical reaction or participates in intricate networks of signaling, its structure emerges through the same quiet process. Order is not tailored for each task. It is universal, adaptable, and reliable. This universality allows life to build complexity layer by layer. Proteins fold into stable forms. These forms interact to create pathways and structures. Those structures integrate into cells, tissues, and organisms. At every level, order emerges from interaction rather than instruction. Folding sits near the foundation of this hierarchy, offering stability upon which everything else rests. There is also a temporal elegance to protein folding. It does not demand immediate completion. It unfolds over time, allowing intermediate states to exist and resolve naturally. This patience ensures resilience. Systems that allow gradual adjustment are less likely to fracture under change. Folding embodies this patience at the smallest scales. As rest deepens, it may be soothing to reflect on how much of life depends on this quiet order. Heartbeats, breaths, and thoughts all rely on proteins maintaining their shapes. Yet none of these processes require awareness of folding itself. The order is so reliable that it fades into the background, supporting experience without drawing attention. This quiet support is perhaps the greatest mark of elegance. The most effective systems often operate unnoticed. Protein folding does not announce itself. It simply happens, moment after moment, sustaining the conditions necessary for life to continue. The elegance of molecular order also lies in its efficiency. Folding does not waste energy. It moves towards stability naturally, guided by the landscape of interactions. Once folded, proteins require minimal maintenance to remain functional. They persist through countless interactions, contributing steadily to the cell's activity. Even when folding varies slightly, order is not lost. Systems exist to guide proteins back toward balance or to recycle them gently when needed. Variation is absorbed without disruption. Order proves flexible rather than brittle. From a broader natural perspective, protein folding mirrors patterns seen throughout the universe. Stars form from gravity and motion. Crystals assemble from repeating units. Weather patterns emerge from temperature and flow. In each case, order arises without a director. Folding belongs to this same family of phenomena, linking life to the wider physical world. As this part draws toward a close, the image that remains is one of quiet coherence. Within every cell, proteins fold and settle, contributing to a stable yet dynamic order. This order does not resist change. It accommodates it, adjusting gently while preserving function. In moments of stillness, it can be comforting to know that such order exists within. Even as conscious activity slows, molecular order continues uninterrupted. Folding persists, interactions remain balanced, and life sustains itself through calm reliability. Protein folding, then, is more than a biochemical process. It is an expression of how nature organizes itself, favoring balance, patience, and cooperation. Through this quiet elegance, life maintains continuity across time, unfolding gently at scales too small to see, yet close enough to sustain every moment of rest and wakefulness alike. 20. A quiet molecular conclusion. As this long and gentle journey through protein folding comes to its close, it is fitting to return to stillness. After moving through chains and shapes, environments and energies, helpers and balance, what remains is a sense of quiet continuity. Protein folding is not a dramatic event, nor is it something that demands attention. It is a background process, steady and reliable, unfolding moment by moment in every living cell. Even now, as the body rests and the mind softens, countless proteins are finding their shapes. Some are newly formed, just beginning their slow settling. Others have been folded for hours, days, or longer, holding their structures with calm persistence. Still others are nearing the end of their time, gently giving way so their components can be reused. This cycle continues without interruption, sustaining life without awareness. What is striking, when viewed as a whole, is how little effort is required. Folding does not depend on force or urgency. It depends on compatibility. Amino acids interact according to their nature. Water shapes structure through preference rather than pressure. Energy landscapes guide motion without direction. The result is order that arises naturally, without command. This order is not rigid. It allows movement, adjustment, and change. Proteins breathe at the molecular level, shifting slightly as they interact with their surroundings. This flexibility is not a weakness, but a strength. It allows life to respond gently to a world that is always in motion. Throughout this exploration, one theme has appeared again and again: balance. Folding seeks balance between attraction and separation, between stability and motion, between order and freedom. When balance is found, function follows naturally. When balance drifts, systems respond quietly, restoring harmony over time. There is something deeply reassuring in this pattern. Life does not depend on constant correction or perfect execution. It depends on tendencies that favor stability and systems that allow return to balance. Protein folding embodies this philosophy at the smallest scale, offering a model of resilience built from patience rather than control. As attention softens further, it may be comforting to imagine the interior of the body as a calm, molecular landscape. Cells glow softly with activity, not frantic or loud, but steady and rhythmic. Proteins fold, interact, and rest, each one contributing its small part to the whole. Together, they support breathing, warmth, repair, and rest. Nothing in this process asks for awareness. Folding continues through sleep, through stillness, through every quiet hour of the night. It does not need encouragement. It does not tire. It simply unfolds according to principles that have guided life for billions of years. This constancy offers a gentle perspective on rest itself. Just as proteins settle into their stable forms without effort, the body can settle into rest when conditions allow. There is no need to force relaxation. Given time and safety, balance emerges naturally. Protein folding reminds us that complexity does not require tension. Intricate systems can arise from simple rules applied consistently. Life builds itself not through struggle, but through cooperation at the smallest scales. This cooperation continues, even when consciousness steps aside. As this final part draws toward completion, there is no conclusion to reach in the usual sense. Folding does not end. It continues quietly, long after this narration fades. Proteins will keep forming, settling, adjusting, and renewing, sustaining life in ways that never need to be noticed. What remains is a quiet appreciation for the unseen. Beneath every moment of rest, a vast molecular order persists, calm and dependable. Protein folding is one expression of this order, a reminder that life is supported by processes that favor balance, patience, and gentle resolution. And so, as awareness drifts and rest deepens, the molecular night continues undisturbed. Within every cell, proteins are folding into shape, guided by the same quiet principles that allow sleep itself to arrive. Nothing needs to be done. Everything essential is already underway, unfolding softly, moment by moment, in perfect calm.